Finally, getting to the sequencing. We've been talking about sequencing for like 4 or 5 lectures, but we still didn't do any sequencing. So today's lecture, I'm actually going into the actual sequencing. That was done for the Human Genome, but I'll be basically talking about the 2 projects quickly, the Human Genome Project and the Celera Genomics approach. And then from there, we'll start taking a look at techniques that are, first generation, second generation, third generation sequencing methods. This is a little bit less confusing because you don't have to imagine contigs one on top of the other and whatever. You just have to see it as a technique being applied to one sequence, and that's it. Okay? Do you have any questions for me? Everything's good? Okay. So the last step before the annotation. So today we're going to talk about how the genome was actually sequenced. So we talked previously about how you break your DNA and put it into these vectors, depending on the size, etc. And then you have to map it by using contig assembly, and that is using the fragments that are made from the insert in your vector. So your initial fragment, you fragment it even more to kind of make a map of the contigs, because you want to isolate a small piece and then go on from there. The step 3 is omitted in whole genome sequencing. This is the shotgun sequencing. This is the approach that was used in Venter's project. Although it's not really true, 100%, because what happens is that Venter, the reason why they got the sequence much faster, it's because they were using the data from the Human Genome Project. So they were basing it on the contig assembly, but if you take just the approach itself for the work they've done, they don't do any mapping, okay? And then sequencing. Sequencing is what we're going to talk about today. So the technology that was used, so this is the initial technology that was used, and then the lecture will go in a flow where I'm just improving on this technology until we get to what's being used today, okay? We'll do this in 2 lectures. Today we'll be talking about 2 techniques that exist, and then we'll continue on from there. So for genome sequencing of the human genome, Celera Genomics and the Human Genome Project used the same approach, obviously because they took the best technology that existed at the time, and this is called automated capillary fluorescent dideoxy sequencing. So if you break down this word, this little sentence into the technique, automated just means that the inflow of the DNA is controlled by a computer. The sequencing itself, the probes, they will fluoresce and be captured by a CCD camera, which then integrates that signal and converts it into a specific nucleotide. So this is the automated part. Capillary, so if you remember, capillary is just these kind of small tubes that replace the gels, right? So there's 96 of them. That was the top that you could do at the same time. Fluorescent, obviously, this one is clear. You're using fluorescent probes that are bound to your different molecules, your different nucleotides, and then dideoxy just means that you're using the chain termination method, okay? So let's start by the first issue that they faced when they did this. So you start with a fragment that is inside your vector, which is shown in yellow. Obviously, you can imagine here, this is the full artificial chromosome. The green is your insert, and your insert is way too big to be sequenced in a single sequencing reaction. So what happens is that, and you don't know the sequence, right? So what happens is that you need to create primers that target your vector, because your vector, it's sequence. Sequence a little bit. When the DNA polymerase gets off, you have a little fragment. Now that fragment, you can use it to make another primer and then continue like that, okay? As you can see, this is time consuming and it depends on the success of the previous step. And so you had to kind of improve this in a way to make it more parallel and less sequential. So the way you do that is by breaking the inserts into smaller pieces. So you break the DNA into fragments that are 100 to 350 kb, and then you break those into smaller fragments to make your contig assembly, right? And then those same fragments, you might break them again even more to do your sequencing sized fragments, okay? So you can see that this is a lot of work, but basically what this does is that if you take your big fragment and break it into 10 smaller fragments, you can sequence these 10 fragments separately in parallel reactions. So as I said, you can do 96 at the same time. Okay? So the sequencing reaction can read about at this point, right? Can read about 300 to 600 nucleotides at a time. So you can imagine that a 100 kb fragment will never be sequenced in one go. And so you need to break your DNA into these fragments that are about 1,500 bases long. The reason for that is you want to, again, do your contig assembly inside the contigs, inside the sequence fragment, okay? So it's like hierarchical. This is what I mean by hierarchical. And then you reconstruct it the same way we learned by comparing the sequences and finding overlaps. Cool? So if you imagine what we learned in the previous classes and you divide it into two things, right here, the same fragment is broken down in two separate steps. The first one is to make the contig assembly, and the second one is to make this, the sequencing sized fragments, and these are then mapped on the contig assembly. So you'll have two different data sets. One is the order of the DNA in which it comes, and the second one is the sequence within these fragments. And then you overlap this with this. Make sense? Okay. Craig Venter went straight to whole genome shotgun sequencing. So you take the fragment, you break it into smaller pieces. So when I'm talking about fragment is the one that you get. So you get your DNA, you break it into inserts that you put in your artificial chromosomes, and then you go directly to sequencing, okay? Obviously, this one you break it into smaller pieces, so you skip this part here. So the approach is quite simple. If your cloned sequences are small, then what you're going to do is that you'll sequence in both directions. Let's start by that. Instead of sequencing in one direction, you sequence in both directions. This way, what happens is that you'll start, let me just use this, you'll start here and here. If your insert is small enough to be sequenced in one go, you'll have the sequence of that fragment. If it's too big, what's going to happen is that it will provide you the information that then you can use to make your sequence contigs. Oh, coming from smaller insert, right? And I'm talking about contigs in two different ways, okay? So this is important to make sense of it. When I say that this step here is omitted, I'm talking about the contigs that are used for mapping the pieces, not the sequence contigs. So this is two different things, okay? So you have fragments that are used just to map, and then there are fragments that are used to get the sequence together. Both of them are contig because they have parts of them that overlap, right? Here, in this case, they overlap because they fluoresce under, or they appear under the X-ray as being colonies that react to the same probe. In this case here, they overlap because they have the same sequence because you have the sequence of both. Make sense? Okay. So two contigs. We're talking about two contigs. The way the whole genome sequencing works, so Celera's approach is that you don't make same-sized inserts. Instead, you make different-sized inserts, and this allows you to basically have fragments that are fully sequenced, others that are not, and then you build from there, instead of having same-sized sequence for the whole genome project. So for the Celera project, they use 2 kb, 10 kb, and 50 kb as examples. You can imagine this, your 50 kb, maybe this one if it was a little bit smaller, 2 kb, and this 10 kb, or whatever, okay? And because you're sequencing on both sides, then what you end up doing is that you're able to overlap the sequences. For example, you'll see that this matches with this, and so you know that these two are potentially from the same fragment. This is called paired end sequencing, and it's useful for joining the contigs, the sequence contigs, okay? I'm talking about sequence contigs here. So the project had 27 million reads of 540 base pairs. So that means they broke the DNA, those different size fragments, they broke them into 450 base pairs long. This allowed you to be in the 300 to 600 range of sequencing possibility. That gave you a 5.1 times genome coverage. And they supplemented this with some data that was deposited by the Human Genome Project group. Okay, so they took that Human Genome Project DNA and broke it into these fake sequences, these 4 reads, let's say, these fake reads, because you already have the sequence of a huge piece of DNA. So then all you do is you break it in pieces of 540 base pairs, and then you map that along with the data that you produced. So let's say, for example, there is a sequence here that has another overlap from here. Then you'll see it because you have broken down their data into pieces that can be matched with your own data. Does that make sense? Kind of? No? Yeah?
I don't understand what the
Right. So imagine the Celeris approach is breaking the DNA and doing everything by themselves. But then new data starts appearing of the human genome itself. And so what you do is you take that data and try to map it onto your data. Or you map your own data onto that data. Does that make sense? And so you break it into smaller pieces, and then you try to use their own data to supplement your own, so that whatever is missing that they have, you can add it. Okay? This gave you an additional 2.96 genome coverage, and then obviously with this kind of work, they got the human genome. As you remember, I told you that the gaps were only recently filled. The gaps were mostly repetitive sequence. That was kind of hard to deal with. Okay? So that was in 2021 or 22. Okay. One last important information about this is the fact that now I'm talking about one set of DNA, but actually it's not one set of DNA. It's multiple sets of DNA. So if you remember, if you want to have a representative group, a representative genome of the human DNA, then you need to include groups from everywhere in the world. So what you'll have is that you'll have these reads. Haplotypes just means that you're taking the DNA from each person into a haploid state, because you don't want to have the autosomes and all that in duplicate and mess up the whole thing. And then you will try to map these using contig assembly or sequence contig assembly to make your consensus sequence. Okay? Consensus sequence means that sometimes you'll find SNPs. For example, here there's SNPs. And these SNPs might be found in a very small portion of the population. So you'll take the most available, the most frequent nucleotide at that point, and you'll use it as the actual human genome, the consensus sequence. Does that make sense? So if you have, for example, everyone has an A at this position, but then you find 10% of the population has T's, well, you'll say that it's an A. Okay? So not perfect, but this is what it is. Then when you have these consensus sequences, you'll do what I just called the paired end sequencing. And because they have the same ends, right, then you can overlap them, right? Because they have the same ends, so that means this end here is the exact same as this one, so then you can join them into one specific fragment. This is called scaffolding. So you're taking different consensus sequences based on their ends, and you're making a scaffold of that. Sometimes you'll have gaps. These gaps are usually due to these repetitive sequences, and so you can pretty much put anything here. So they create these gaps which were filled recently. Okay? Then when you have these scaffolds, you can then map them using those sequence tag sites. So remember, there was a map of about 30,000 or 40,000 tags. So then you can use that to help you and map those consensus sequences along the chromosome itself. Make sense? Yeah, okay. So Craig Venter started by using his own DNA and then ended up using the DNA of everyone. So that did not work. But their effort together was successful into getting the human genome much quicker. His approach was much better because it was faster, but it ended up relying heavily on the data that was published by the Human Genome Project. And those scaffolds were anchored to the mapped STS that were done in like 1999. Okay? Good. So just a little bit of speed comparison. Human Genome Project started way before this, but the actual full-scale sequencing efforts, so I'm talking about the sequencing effort, not the contig assembly and all that stuff. I think it took them like 13 years, right? So the biggest part is, if you remember in that 1 to 5 from genome characterization to annotation. So if you take steps 1 to 4, steps 1, 2, and 3 took about 10 years, and the rest took about 3 years. Okay, 2 years, sorry. Make sense? So the draft sequence was published in Nature Science in 2001. Now, obviously, you want to resequence the genome, right? Because this was a draft. Now that you have more sequence available, you can actually retry at different positions and things like this. And you can make primers at different places and then try to map that based on those primers. So in 2003, it took 3 months using 100 sequencers. Now it takes about 5 hours to do the whole human genome using the Oxford nanopore technology. And this is actually in the Guinness World Record book. I don't know where they're doing this, but scientists got bored or something, you know. If you can do it faster, maybe you can win something, you know. So what we just talked about, that's first-gen sequencing. That's the first ever available. Now, there's every few years, there's a new generation that comes up. So at that time, when we talked about next-gen sequencing, we were talking about second-generation sequencing. Now, when we talk about next-gen sequencing, we're talking about fourth generation. That's where we are right now. And the main difference is that you're basically doing more reactions at the same time. So you can sequence millions of reads instead of 96 reads. Okay, so that's the first thing. They usually had an amplification step. So what you do is that you'll do a PCR reaction to increase the amount of DNA. And that allowed you to work with much smaller volumes of reactant, make less reactions in your sequencing steps. Generation 3 technologies got rid of the amplification, and obviously 4 are now taking like one piece of DNA, and that's it. That's what they're doing, okay? Usually, there's 3 methods. So the way you do this in second generation sequencing, either through synthesis, so this is what we learned about, the dideoxy, or ligation. So you're adding, you're ligating probes sequentially until you get the full signal. The only one that does not do that by synthesis is Oxford nanopore. And if you remember in the presentation I showed you at some point, I think lecture 4 or 5, the way Oxford nanopore works is that the piece of DNA goes through a pore, and the electric signal that comes out from that movement is specific to each nucleotide. So as the nucleotides come in, they'll generate a signal based on the physical properties of that nucleotide, which is different between A, T, C, and G. Okay? Today we'll be taking a look at 2 technologies, the Roche 455 pyro sequencing, and then the solid sequencing from Applied Biosystems. Okay, Applied Biosystems is the same technology, the same company that provided the technology for the HGP and Celera's approach. Okay, so they improved on it by creating solid, and now they're killing it in the market. You'll see why. So now you don't need to clone fragments anymore in E. coli to amplify it. You can do it, but you can also just do a PCR reaction. So much more faster. You have a lot more parallel reactions happening. So again, another improvement. And one last improvement is the amount of reagents that you use relative to the data. Okay, because you can reuse some of these reagents for your reaction example, the probes or things like this. And so this is one of the main things why the price has decreased a lot. The other reason is this cloning here. That is quite annoying. Okay, so this is what I just said. Decrease cost, increase speed. The Human Genome Project took it 13 years to do, $3 billion. Now, third generation platforms can do it in hours, about $1,000. Actually, it's $249 to do it. I'll show you this in a second. But we say that we decrease the price of sequencing for the whole genome to $1,000, but in reality, you need $10 million machines to do this. So it's not really $1,000. It's more like drop the 10 million, get the machine, and then it's going to cost you $1,000. Okay, but I guess it works. So you need a lot of sequencers at the time. Now you only need one, and it's about this big. Okay. Nebula sequencing. Who knows this? Okay, so actually, you know what? Let's go on the website. Pretty sure some people are gonna explode their money on this after today, but I'll show you this. So nebula sequencing does genomics. It sequences your whole genome. Very user-friendly platform. So you can click on Decode My DNA. They will send you, a kit. You give them your DNA. They have a mobile app, and that mobile app tells you everything about your DNA. So they go through, that little star here is funny, but they go through most of your DNA, obviously. Let me see, where is it? Pricing there. Pricing. First thing, you know what a genome coverage of 30x means right now, right? So this is the first thing that you know. So this is what they mean by deep coverage, is the fact that they do a genome coverage of 30 times. So this is highly precise and covers most of your DNA. As you know, the Poisson distribution will never reach 100%. And so this is why you can increase up to hundreds of genome coverage, but you'll never get perfectly the full genome. So you need to do different approaches to get the full sequence, okay? Because 99.999999999 is never 100%, right? But this is pretty big. You pay them $249, I don't know if it's Canadian or US, and they give you all your DNA. They tell you which genetic mutations you have. They'll tell you what predispositions you have and where you come from, okay? This one here, the only difference is they increase the genome coverage. I think in this case it's like 100 times right here, okay? I'm not getting paid by Nebula, so I'm just showing you this. If you're ever interested, it's pretty cool. But yeah, so now it's crazy because before this was not available to anyone. Now you can just go on there, pay a little bit of money, and then get your whole genome sequenced quite quickly. One thing though is, I forgot which company used to do sequencing. Then they got bought by their investment firm. So you signed this disclosure agreement with that company. Then the company gets bought by their investment firm. Those don't apply anymore. And so data from millions of people that went through that company's sequencing approach, their data ended up being unprotected in that firm. I think it's 23andMe or something like this, okay? So you have to be careful with this if you care about your genomic data, let's say. I guess you can send it under a different name, You can find a way, right? Just buy, those credit cards that you get from Pharmaprix and put some random name, So if you're interested in this and you want to learn more about this, I put videos for all these technologies. You can watch them. It's always more interesting to take a look at the videos because they're animated, and so you can see what happens, and some of them are somewhat complicated. So if you don't see the animation, it's a bit hard to imagine, but at your level, I think you should be able to do it. Cool? So let's test your imagination, okay? Start by two techniques. First is the 454 pyrosequencing method. This was the first successful massively parallel platform. It was designed by Roche, which is one of the companies that you guys probably know, and it was the first commercially available. So 454 is the light detection wavelength, if I remember correctly. Pyrosequencing refers to the fact that you're creating these pyrophosphates. Everyone knows what a pyrophosphate is. It's two phosphate groups bound together. So one phosphate is phosphate alone, and then this is diphosphate, or you can call it pyrophosphate, okay? And it supports a sequence read of 450 nucleotides. So this has decreased a little bit from the previous method, but it allows you for this massively parallel sequencing approach, which allows you to do 1 million reads per plate instead of the 96 that was done with Applied Biosystems 3730X whatever. Okay, so this is the system that was used for the Human Genome Project. Not one of them, but hundreds of them. What else? And it does not use fluorescence, it uses light emissions, right? And we'll see this in a second. So basically you have your DNA molecule, you put these nucleotides, DNTPs, they get attached to the sequence, and then as it's building this, there are pyrophosphates that are released. These pyrophosphates are used by an enzyme, which we'll see in a second, to produce ATP. And this ATP is used by an enzyme called luciferase, and luciferase, it basically makes light. Okay, it produces light which can be detected. So let's go step by step. So the Roche 454 sequencing method goes as follows. Imagine you have your DNA right here. You will break it into pieces that are about 800 base pairs long or less. You'll add these adapters. Two different adapters, A and B, and these adapters are important because they will be used to bind to a bead. So this little piece of magnetic bead that will have the opposite sequence. So if, for example, you have an A adapter, then you have the complementary sequence for the A adapter to bind. So you'll put your complementary to A sequence here, and that will get the DNA that is bound by this adapter. Okay? Then you separate your DNA into single strands, and you'll try to immobilize only one DNA molecule, one single-stranded DNA molecule, onto each bead. How do you do this? This is just a question of concentration. So you have to make sure that you're playing with the concentration of your beads and your DNA molecules so that you end up immobilizing only one DNA strand per bead. Sometimes you'll have two, sometimes you'll have three, sometimes you'll have no, obviously, but you try to get most of your population to be at a 1-to-1 ratio. Okay? Then, this is where I need you to focus. So your sequencing, so let's start by the buffer. Your reaction is happening into some sort of oil. And inside the oil, you have these beads that are solubilized into water. And so when you put these beads inside the oil, it will create these droplets that envelop the bead. And they will envelop it just because of the way the lipids react with water. So these micelles, do you guys remember what micelles are? Okay. They will encapsulate the reactions, so they will make kind of a small compartment into the oil, and the lipids from the oil will kind of be around that little water droplet, which contains one bead. It also contains reagents to do the PCR reaction. So your primers against the adapter, the other adapter on the other side, because one is, so you have your bead, you have an adapter, you have the DNA that binds to that adapter, and then on the other side you have another adapter, right? So that adapter, you know it's sequenced, so you use it for the PCR reaction. Yes, Julien?
Present a lot of different sequencing methods, just this one, but what's the pros and cons compared to some of the other ones you were? Is it better throughput? Is it better accuracy? Is it better?
Yeah. So I'll be doing pros and cons for each of the method. After I explain the method, actually, I have some questions about that so that I can see if you guys can see any problems with them or any advantages, and then I'll tell you what's good and bad between each one of them, okay? Very good question, though. So in this case, what happens is that you'll have a PCR reaction inside that water droplet that will amplify your DNA molecule, and it will stay within that water droplet, and then because there are adapters on each side, that will be bound to the bead. And so you'll have a bead with hundreds of different DNA, of the same DNA molecule bound to it because of the PCR reaction. Does that make sense? Okay. This is called emulsion PCR. Emulsion just because of the lipid water droplets and PCR polymerase chain reaction. Why would you do this? This is just to increase the signal, because one molecule is not going to produce enough signal to be detected, so you need more molecules to kind of do that. Yeah, Matthew.
So the reason you're doing it in an emulsion is so that you keep the identical sequence on each bead?
Exactly. Yeah. Let me just start writing names. So Julian, Matthew. That's it, I got it. Okay, after that, what you will do is you'll do a step of pyrosequencing. I'll explain this in more details, and you'll detect the pyrosequencing using some chemiluminescent signal. So light produced from chemical reactions. Okay. I just explained this. You can reread it. I just put it here for you guys. It's basically what emulsion PCR is. The beads are Sephiroth beads. You have oligos that are complementary to the A and B linkers, which I just told you, the adapters. They're bound covalently, so they don't move from there. If you're doing research ever in the future and you're trying to precipitate a protein, you will use a different type of bead that is bound to an antibody, for example, things like this, right? And so there are different beads that can precipitate different molecules, and then you do your reaction in an oil-water mix with millions of droplets, and you try to trap the same single-stranded DNA in your droplet. Okay? Then you can get to the pyrosequencing. So pyrosequencing is where you actually start getting the sequence from each of the fragments. So imagine a plate that has 1 million small holes like this. Okay? The beads is this yellow thing here. Right? So there's 1 bead per well, or per capillary, or whatever they're using here. It's a well. And you have 1 million of different holes like this, okay? This is called a pico-titer plate. So it has millions of different spots like this. And in each one, you try to get 1 bead per hole. This is the yellow thing here. And all these DNA fragments that you see here are the exact same because they were created in a emulsion, let me just remove this, in an emulsion PCR step, okay? So that's that. Now, because you have an adapter on the other side, so you have 2 adapters, one that is for the bead and one that is for the primer that will be bound here. So you'll need a DNA polymerase, you'll need your primer, and you'll need also 2 enzymes that you also put on different set of beads, the sulfurylase and the luciferase, and I'll tell you what these 2 do. And then you also need adenosine 5-phosphosulfate, right? I'll tell you what this does also. But basically, your ATP sulfurylase will take APS and turn it into ATP, okay? And I'll explain this in detail, so I'm just telling you the components so you get an idea. You need, obviously, luciferase, the enzyme that makes light. You need the reactant, or the molecule that will make light, okay, by being transformed by the luciferase. And then you need another enzyme called a pyrase or a pyrase or whatever, which degrades nucleotides, okay? So this is all what you need. Now, how do you actually do the sequencing? So you mix all the components in each wall, except for the DNTPs. The DNTPs, you have to add them at each one, like you have to add them one by one. So what you will do is that you'll add the DNTPs, you let them react, light will be produced, then you flush those DNTPs, and then pass a second set of DNTPs, Gs, and you keep doing that in intervals, okay? So you'll have A passes, G passes, C passes, T passes. Whenever you see light, you repeat the cycle, okay? And you can do this basically for each of these wells, what we call wells, or these holes here. So the light is detected separately for each one of them. So you can see how parallel this is. Then, whenever the nucleotide, so you have your primer that binds to the adapter, which you had at the beginning, this guy here. And then when the reaction goes through, so then if you put T, and then T is the one that's supposed to bind because you have A on the other side, then there's a reaction that happens which will release this pyrophosphate. This pyrophosphate is a reactant that is used to then produce the ATP, which is used by luciferase to make light, Okay, so ATP is the energy required by luciferase, and the only way to get it is through this pyrophosphate. Okay, the amount of pyrophosphate released is basically proportional to the amount of nucleotide incorporated, because you can have multiple T's that go in a row, right? So you need to detect that. So if there's 3 T's that are added in a row, you need to differentiate between 3 T's and 1 T being added, and that's just the amount of pyrophosphate being released. Okay. My question for you: how do you differentiate between each of the DNTPs? You're adding them at different times. So first you have the DATP, then the DCTP, GTP, but they all produce the same pyrophosphate. So how do you differentiate between them? Yes, Alex.
Really need to, if you know which one you added, because it'll just create the light.
That's it. I was trying to trick people, but yeah. So basically, because you know which one you added, then the light that is produced is related to that nucleotide and not another one. Okay? Was that clear, or some people had to think? Yeah.
I have a question. Because it's in water and you do the PCR, you have to change the temperature. Doesn't it kind of like
Affect the lipid droplet? No. Oil is pretty resistant to water, to heat, and depending on which oil you're using, then it will not be affected much. Okay. I forgot which ones are more sensitive to. I think it's unsaturated fatty acids that melt, and then saturated are just oils already, and so you can heat it up, but nothing's going to happen. Okay. Anyways, the amount of heat that you're adding to do the PCR reaction is not to the point where the oil will, light up and make a fire. Okay. Because usually the burning point, or whatever it's called, for oil is higher than the water melting, whatever. I don't remember, but makes sense. Just Google it, okay? So you can figure out the different temperatures. Okay. Now the light stuff. So here I need you to focus so you can understand why we're adding this APS. So you don't want the ATP, the DNTP, so the ATP, DATP, to actually be used by your luciferase enzyme to produce light. So you have to differentiate it when you're putting the A into the reaction mix, so this A that passes here, right? You have to use a different A. And this A is called, wait, what am I saying here? APS. Yeah, exactly. You have to use APS to produce the ATP. APS will be used by sulfurylase to remove the thiol group, so the sulfuryl group from APS, and add this phosphate here to make ATP. The sulfate group is removed, and then ATP is added. Okay, chemistry is a long time ago, but I still remember. Okay, so let me repeat. Sulfurylase is the enzyme that will take the pyrophosphate to produce ATP, which is the energy source of the luciferase reaction. To do that, you can't use ATP, the DATP, right? So you have to do 2 things. The first one is add APS, which can be converted by this enzyme to ATP. But then this ATP, you want it to only be used for the luciferase enzyme reaction, and the DATP, you want it for the actual incorporation. So you need to use a special DATP, which I'll show you in a second. But basically, your ATP will then be used by luciferase to convert luciferin in oxyluciferin, and that makes light. That generates light. Okay. Now, how do you differentiate the ATP that's being used for the luciferase enzyme and the DATP that is being used for the incorporation? You need to use a special DATP, which is probably next slide. Yeah, so I'll talk about that in a second, but the light that you're producing is proportional to the number of nucleotides incorporated, as I just said. So that means If you have, for example, 2 or 3 A's that are added in a row, you'll produce 2 or 3 times more light. Okay? Let me ask you a question. What's a problem with this technique in terms of light? So if you have 30 A's in a row, can you detect 30 A's? Yeah. The light? The light is just measured by just these cameras, these CCD cameras that actually detect this light. So I don't know if you guys are aware of Western blot or things like this. The camera just basically detects these photons, and that's it. Sorry? Is it reliable? It is pretty reliable. But wait, let's try here. Yes. Sorry? If a mutation does what, sorry? Yeah. If a mutation happens in the polymerase reaction, like an incorporation of the wrong nucleotide, can that even happen? Let me think. That's a very good point. I never thought about, the wrong nucleotide being incorporated in this case. I would assume they do these reads multiple times to confirm the nucleotide that has been incorporated, because the polymerase that's used, the clino polymerase, does not have a repair mechanism. So it does not go backwards, check if it's the right one, and then go forward. But that's actually a good point. Yes, Alex?
It doesn't know to stop at one nucleotide, right? So if you had 10 T's in a row, it would just burn through all of the T's, wouldn't it? So you wouldn't be able to find the homopolymers very easily?
It would. So you're telling me basically that it adds those A's and just burns through them and does not.
Yeah, you could try to correlate it with the amount of light, I imagine, for that cell. But
Yeah.
you would lose a lot of accuracy. You'd be terrible at having a long string of the same base, I would imagine.
Actually, it's somewhat precise up to a certain point, but you're close to the answer. Yes.
Maybe it has to do with the detection, the just noticeable difference between 29 A's and 30 A's isn't enough.
Right. So this is the actual answer. So there's only so much light you can detect, right? At some point, your camera is saturated, and then all you see is basically the same signal. So first you see the signal increase, but at some point there's a limit, right? And it's same for us. If there's too much light, you just don't see anymore, right? And so this is the same thing with this technique. One of the problems is that after 5 or 6 nucleotides, you basically that are the same, you basically don't detect any more higher than that. And so what happens is that the way you fix this is by having your different segments and playing with those repeats to kind of break them. Okay, so that's that. So let me just write. Sorry, what's your name already? Heidi. I always forget. Heidi, and then Gabrielle. So I have Alex that answered, Gabrielle, and Heidi. Anyone else I forgot? No? Okay. For this question, I mean. Okay. How do you then avoid the DATP being used by luciferase, because it can use it? You have to add a special type of ATP. So this DATP alpha sulfate, right? So this basically thiotriphosphate, and it basically has a sulfate group or a thiol group that is added to the alpha phosphate in all the 3 phosphates. Okay, so this one is used to replace the ATP. Okay, easy, good. One last step. Every time you put a nucleotide in here, you will have to remove it whenever the reactions are done. And to do that, you have to use an enzyme called apirase or apirase or whatever. And it takes just the DNTP and breaks it into the DNDP and the NMP, so monophosphate, diphosphate, triphosphate, and releases the phosphate groups and not the pyrophosphates, because it breaks one phosphate group at a time. Okay? This is a nucleotide degrading enzyme, and it does the same for the ATP, because some ATP will be left that has not produced light, and you have to obviously normalize using the machine for this. And so whenever the reaction is done, after a certain time of capturing the light, then you have to clean it up and repeat. What else? Can you see a problem with this? Yeah, Arthur?
Not a problem. Is it what we do when we have too long sequence of the same nucleotide, or is it different? When you say we play with the repeats, when it's too long?
Oh, the type of fragments that you produce here. So because you have a DNA this big, then you'll break it into 1 million pieces, which you'll put into this picotiter. So then you have to kind of try to avoid those, because you'll not get it 100% in the first run. That's why you need a higher genome coverage and all that, to have different variations of the cuts, right? And then you try to avoid these repeats, right? But obviously, at this stage here, that was not the perfect technique. So this is one of the problems with it, and it was fixed in third and fourth generation sequencing. But this is one problem that used to exist, and they still use this technique even though they had that problem. Okay. What else? So this is the signal I was telling you about. This is a pyrogram. So the intensity of the light tells you how many nucleotides are added, because you can basically have a standard curve for each of the lights, and you know that this is one nucleotide being added, while this is basically 2 or whatever. And there's a limit at the top. Above this, you don't see any more change, so it's saturated. The major problem is this one here, is these homopolymers that look like this. So after 5 or 6, you cannot distinguish anymore if it's 7, 20, or 30 in a row. Okay. Poly-T-tails, right? So your poly-A-tail, poly-T-tail, those are big problems for this technique. Okay. Good, not bad. Doing better on the timing. The second technology is part of the, it's called SOLID, and this was created by the company Applied Biosystems, which had the first technique, the first generation method that was used for the Human Genome Project. So obviously they tried to get into the market again with this method, and this one here is not through synthesis, but it's through ligation. And the difference is that you're adding at the end of the DNA instead of polymerizing, right? So that's that. This one was a little bit better than the other one in terms of ensuring that you have the right sequence, because it used a detection system that detects 2 bases at the same time instead of 1. And it used it walked back on the DNA, and then you would have the 2 different bases being, so let's say, for example, you have 3 bases. You'll start sequencing, let's say, these 2, this one and this one, and then you'll go backwards, and then you'll sequence this one, and so you know that the middle one is exactly that, okay? So this method here, this is what SOLID refers to, sequencing by oligonucleotide ligation and detection, because your probes are oligonucleotides of 8 nucleotides specifically. 2 of them are specific to your bases that you're sequencing, and the other 6 have the probe attached to them, okay? So the way this works, follow a little bit so I don't lose you. You have your DNA, we'll denature it, just like we did previously. You'll attach these adapters at each end. You have adapter P1 and adapter P2 instead of A and B, but basically the same thing. And then you're going to attach these using a complementary sequence to one of the adapters attached to a bead. And then you will extend from the other side, because you have the sequence of the other adapter, and then you'll extend from there. But you will not extend by doing, you'll be ligating instead of synthesizing, let's say, okay? This here, I just realized that there is kind of a mistake here. So I used this diagram because it showed what was happening, but this polymerase here, remove it. Okay, is that clear? Just this part here, forget about it. So you're extending on the other side, but you're not extending through a polymerase, you're extending through ligation of your probes. And basically you repeat that nonstop until you have enough signal. So clonal bead population just refers to one bead that has the same clones attached to it multiple times. Obviously you have your water droplet, which contains the template, one single template, which is amplified through emulsion PCR, primers, beads, all that stuff. And let me move this stuff here. Jeez. God. Okay, let's just leave it here. There you go. One difference is that after you produce these beads that have the amplified DNA and all that, you will attach them to a glass slide, which looks like this. And you can attach them into different conformations, 1, 2, 4, 8 chambers, whatever you want. This is just basically how parallel it is. To do that, the end of your sequences, you will add to them, you will basically modify them so that they attach on the slide. And this is what will be kind of sequenced, okay? Each piece by itself gets sequenced. Each little component gets sequenced on the slide. Okay, this one is a little bit harder to understand, so try to focus a little bit. So the way this works is that you have your P1 adapter, you know its sequence. So you're going to start, you're going to put a complementary sequence on the other side to which you will ligate different probes based on these two nucleotides here on your template DNA strand. So at first you will start exactly, so your probe will be exactly at the same position as the end of your adapter. So your primer, sorry, not probe. So you start at the same position. Then what you're going to do is that depending on these bases here, you will be ligating different probes. This is the 8 oligonucleotide probe that I told you about. In this case, it's the AT probe that binds to TA, okay? And it makes red light. If you had a different one, let's say, for example, GC here, it would have a different light, okay? Then you will excite the DNA molecule, the probe basically, and that will fluoresce and produce color that you will detect in the computer. Then you will break the probe, and by breaking the probe, you don't break the full, the piece, the remainder 6 nucleotides that are not bound to anything. Instead, you will remove 3 with the probe, and you will leave 3. So this will create a space between each probe binding. So then again, you bring it another probe, and that probe will bind 3 nucleotides down from the first 2. Do you get this part? Kind of? Sarah? Yeah. To the parts, the middle parts? Yeah. We'll basically have to do this 5 more times, and we'll move the thing. But I'll get back to this. I just don't want to mix you up. So then you'll add a different probe. That probe will come and bind right here. It will produce, let's say, blue light because this one here is blue, and the combination of nucleotides is TT. You break that probe. You leave 3, a spacer of 3. Do the same until you finish your DNA molecule. So when that's done, now you have that sequence which has holes in it. So then you have to take your initial sequence and repeat the round, but in this case you will take your initial primer and you'll shift it by 1 base. And so what happens is that you will sequence again the full sequence, and that will give you an additional nucleotide in here. Then you shift it another nucleotide, and then it will give you the second missing nucleotide, and a third time it will give you the third missing nucleotide. So you can see how this system allows you to sequence the same base multiple times, and this gets you to actually have a very accurate sequence. Does that make sense? Yeah.
There is only 4 probes or more. There should be 16.
No, in this example here, I'm just showing these guys, but there's a lot more.
Okay.
Yeah, this is just this picture here has this here, but they're using 4 different colors only. So we'll see how you can differentiate between the binding, because technically you'd have 16 probes, but they have only 4 colors. So then that means the same thing will, so different combinations will produce the same light, the same color 4 times. So let's say AT, TG, CG, and whatever, they will all be red. So then we have to see how you can figure out what the hell is there. Okay? Yes, Alessandra.
So basically, in each cycle, they introduce all of the probes, and then they see which light it gives off.
No, they add 1 probe at a time, excite, repeat, cleave.
Then how do they know which probe to insert?
Sequentially. They just one after the other, just like
They insert one, and if it
binds.
if it binds, it binds. If it doesn't, they just try another one.
If it binds and then produces this light, then that's it. If not, then they will wash and then repeat. Okay, and it uses a capillary system, same way. This one can do also a million different reads at the same time. Okay. So it seems like it's a lot more work, but actually, for you to be so the other one, the first generation, what you're doing is that you're adding 1 nucleotide at a time and running it through a gel or capillary, and then you get different fragments of different sizes, and then at each time you detect the light that is produced. It's kind of similar to this. The only difference is that this allows you to do them in a parallel way instead of sequential way. So this is why this seems to be taking more time, but because it's in a parallel fashion, it's less time. Okay. What else? So this is what I meant by you move your primer that binds to the adapter. First, you start at nucleotide number 1, then you go backwards and then forward, etc. So you'll be covering the first 5 nucleotides using your primer, and then from there you'll get different combinations of color, right? And those different combinations of color will tell you which nucleotides are there. And I'll show you that in a second. If this mixes you up, there's a video here. It took me some time to understand this the first time. So if your first base is T and the second one is A, you make red light. If your first base is G, second one is C, you'll make red light. CG makes red light, and AT makes red light. So then how do you differentiate between these 4? And this is where you have to know your first nucleotide. And you know your first nucleotide based on the primer that you're using for your adapter sequence. Okay? So what you do, because your initial read, you read from number 1 and not from number 0, you're basically you're allowing for 1 nucleotide of the primer to be read along with whatever is there. And so you know that nucleotide. Now, because you know that nucleotide, then if you get red light, right, then you can think that because you know it's A, the first base, right, and then you get red light, then you automatically know it's a T and nothing else, because you know this A from your primer that is ligated to the adapter, which you have shifted by 1 base. So the first base, you know it. Okay? So that's the first thing. And then you will go 1 step further. Then you'll see that you have a green light. So you know that your first one was AT. So that means for this dinucleotide that you are trying to sequence, the first one is T. And you got the green color. So T is your first nucleotide. Green, that means the second nucleotide is G. And then you keep doing this until you get the full sequence. Ça fait du sens? Works? Simple. So it's very, it's not really complex. It just takes time to go through, but the computer does this for you. All you need to do is get the first step properly, okay? You don't need to think about what nucleotide was there and all that. You just put what was your primer that you used, the adapter that you used, and then it will tell you whatever there is right after that. You can also read this review, which tells you about this technique. So it's pretty ingenious. One of the advantages of this is that it's highly accurate. Can you see a disadvantage of this? Yes, Maria. Yeah, so it's a very good point. We ran away from the sequential, but we're getting back because you're doing reads from different positions, right? So you have to read kind of this and then this and then that and that and that to get the full sequence. There's no way to do it in a single run, okay? One thing that's good about this, though, compared to the previous technique, anybody has an idea? So what was the major problem with the other one? Sorry? Yes, that's one thing, but in terms of the, yes, go ahead. Right, so this solves the problem of the repeats, right? Because if you have repeats in there, you'll be producing different colors depending on the repeats, and because you're doing it multiple times backwards and forward, it makes sense. You're not reading 6 repeats. You're not getting light from 6 repeats at the same time. You're getting it only from 2 each time. So yes, Julian.
If you're also doing it, every base pair basically gets scanned twice because
Yeah.
of the way you work through everything. So does that like decrease the false positive rate?
Does that decrease the false positive rate? Yes, of course it does. I have a slide on that in a second. Okay, I'll tell you about the false positive reads because let me just write the names: Julian, Gabrielle, Heidi, and Maria. Anyone else? No. Okay, good. Where was I? So I'll talk about the false positive reads in a second. So one cool thing about this technique is that you can use it to spot single nucleotide polymorphisms. So you can use it for de novo sequencing. So that means you don't know the sequence and you're sequencing it. But let's say, for example, you're looking at the CFTR gene for cystic fibrosis, and you want to see which mutations are found in this patient that basically lead to that person having cystic fibrosis. So you know already what the sequence of the healthy cystic fibrosis CFTR gene. So that's called your base space reference. Then from this, you can make your colors, CG red, GT green. So this is your base colors from the reference sequence that you already know. Then what you can do is you can go and take the patient's sample and sequence it with the mutated CFTR gene. And so that will give you reads like this. Now, why do you have 3 rows? Because you'll do it multiple times. In this example, it's only 3 rows, but the number of times you sequence it is multiple times to make sure that you have the exact sequence. And depending on the fragments you have, maybe this fragment will start here, the other fragment will start here, etc. Okay? And so in this case, you have 2 fragments, for example, in this sequencing run. This is one of them, and this is one of them, and you're missing a piece here. Okay? So then you'll do your reads using this method. And let's say you have 3 reads, then you'll try to find your consensus sequence from these reads. You have to make sure that you have one line of colors from all your different reads. So this is kind of your context, let's say. Okay? So here you have red, green, and then here you saw red in 2 different samples. So that means this one is red here. Then from there, you can extrapolate the sequence, the base sequence, right? One thing that you have to realize here, though, is that your base sequence has these colors here, right? So you have red, blue, green, yellow, but in this case you have red, yellow, red, yellow. So obviously there's something that changed here. And because there is a mismatch between the base sequence and your patient's sequence, then you know that there's a point mutation here. Okay? So this is very good for finding mutations. If you take samples from thousands of patients, you can find all the mutations that potentially lead to cystic fibrosis, for example. Obviously, some of them are not going to cause cystic fibrosis, so you would have to take a healthy sample, mutate only that nucleotide, and then test it in mice or whatever and see what happens. Okay? Good. So this is that. Let's see. Now, SNPs, so if, let's say, for example, you have your reference sequence, which is here, and then you have a read here that has an SNP, you'll see that only 2 colors will change. So when you see 2 colors changing, you know directly that this is an SNP, right? If you have your base sequence that comes up, and then, so you have your base sequence, you compare it to the read of your patient sample or whatever, and you see that only one of them changes. This is probably due to a false positive and something happened here. And you're detecting only one nucleotide change, but in reality there isn't any, so you have to fix this thing here. So this is how they troubleshoot this method. Because it is a 2-base system, you usually expect 2 bases to change at least. If you have a deletion, this will appear as basically just the colors shifted, and then you can kind of rebuild it using the computer. So you'll have the same colors on this side, and then you'll have basically this here that's missing, so that's your deletion. If it's an error, you'll be producing a color that is totally wrong. Okay, so this is the difference. Insertion, you'll be adding, so you're adding a nucleotide, right? And so you'll basically be adding one additional color to the sequence, okay? 2 different colors, sorry, not a single color, because it's a 2-base system. So if you're adding one nucleotide as an insertion, that means you'll be having 2 colors that change, okay? You can take a look at this and think about it a little bit more thoroughly, but this is how you can go through the data. Actually, you don't even need to do it. The computer does it for you, but if you want to understand it by yourself, then you can do this. So I think there's 2 slides left and a little homework. So the first slide is, so here, the advantages of this method here, it's used a lot when you're choosing your method of sequencing, sometimes you'll choose it based on the pros and cons of that method. If you have too much DNA, then you'll use a different method compared to if you don't have enough DNA and you really want to make sure that that one run gives you the sequence and that's it. So there's different ways to do this. So you have to choose which one fits your sample. That's the first thing. And then solid, what it's good for is for these polymorphisms. So because it uses a highly redundant system of 2 calls for each nucleotide, then it provides you more with a higher confidence when you're calling a single nucleotide polymorphism. So if you find something that changed there, it's probably because it changed. While in the other methods, it could be just incorporated by accident, and you don't know. You will not know if it's actually the actual nucleotide that was added, or if it's something that happened due to your actual run, okay? The other thing is that the short run lengths, so when you're doing small pieces, this technique is not so good for it because it does so many reads that you will end up doing more work for no reason. So there are better methods to do this. If you have large sequences that you're trying to assemble de novo, you will not use solid, okay? Because you're basically reading and rereading and rereading the same piece too many times, and that's not interesting when you're doing de novo sequencing. Okay. Homework. Is it today that I'm giving you homework? No? Tomorrow?
The last homework, there was no place to drop it on Moodle.
Yeah. So I moved the deadline to Friday. The reason for that is I didn't have time to finish correcting your assignments, and I want to give them to you absolutely before you submit this one. In general, it's good. One thing that I observed is that some students go, like, their arguments are abstract. So abstract arguments are not sufficient, okay? What do I mean by this? Obviously, the bioengineering conspiracy theory does not make sense for political reasons, because if you're bioengineering a virus, and then the whole world is affected, then where's the bioweapon in this, right? So that's the first part. But in terms of genomics, I need a little bit more specifics, right? Some people, they use politics more than science. It's not bad, but science is important. It's a science class. It's good to supplement the science with politics, but it's also important to remember that it's a science class, okay? So this is one thing. I'll give you an example of supporting your data by science for this assignment. For example, there is a student that talked about, so people say it was not bioengineered because it does not make sense politically, whatever, or we don't have the technology to do this. Actually, we do. If you do a little bit of research online, you'll find that there's another coronavirus that was engineered before this one and was published that it was engineered, and that the researchers from that team that made that coronavirus, in 2020, they published another paper to say it's not us who did this new coronavirus. So this is the kind of science research I need you to do, not just it doesn't make sense because the whole world got affected. Yeah, of course, that's clear, but where's the science behind this, right? So that's one example. This actually, I think one student did both of this and killing it. You can use even philosophy if you want, but as long as your science was there. And I will not tell you the philosophy part today so that we can let those students have their own advantages there. Not just give you the whole freaking thing that they wrote about, but you can even use philosophy. If you search a bit, you'll figure it out. But the science is very important. Some people talked about recombination. Some people talked about traces of genetic engineering. How do you know? So you take the genome. So you're a geneticist or a genomics expert, and your goal is to understand if that DNA is engineered. You have to think as a scientist, not as and I'm not, no offense, I'm just trying to teach so that you understand the mistake. When you say we don't have the technology to do this, that's totally wrong. That's telling me that CRISPR doesn't exist, right? Lentiviral or viral systems for induction don't exist. We don't have like ways to cut DNA. We don't have ways to break the DNA. That's not true. We do have those methods. And so how do you find if it was CRISPR engineered, right? So then your thinking as a genomics expert is to say, well, CRISPR leaves these traces in the genome. And so if it was engineered by CRISPR, then we should find those traces. So this is the kind of thinking that I need you to do, not just we don't have this technology and so obviously we didn't make it. Or it affected everyone, so obviously it's not a bioweapon, okay? Yes, those are arguments, but not in this class. Like, you have to supplement it with scientific stuff, okay? So, yeah, 1 second, Julian. So I will correct everything. I will give them to you tomorrow with enough notes so that you can now understand the scientific thinking behind this. And if I see something that you say that seems off, then I'll ask you a question about it. What about this, right? So I'll just give you one example so now you start thinking, right? So the DNA that was taking, the RNA that was taken from your nasal swabs, it could have been extracted and turned into DNA, right? And from your own cells, and that used to basically have DNA of everyone in the world, right? So how do you disprove this now? See what I mean? So there are many things that you have to think about that maybe don't come right away, but you have to think of the possibilities based on genetics. What can you do with that sequence? What's the advantage of doing that? And what science is telling me that this is not true or true? Okay? Other than that, Julian?
Just a question about homework. When you say the first discovered gene
Yep.
in the autosome, do you mean human genes or do you mean...
So I made the mistake of not saying human, but if you found another organism, that's okay.
I was assuming human because it's more medically important.
Yeah. But since we're doing genomics, I realized that I have to focus a bit less on human and diversify this. So if you found one in, I don't know,
So it's okay to either represent human or non-human.
Another organism.
Both are good.
Yeah, both are good. I will do that. If your gene is around 1960 or 70, then redo the research, okay? You're like 50 years away. So that's that. So Friday you can submit the homework. If you're, I asked you the first gene, but obviously you might find third, fourth, whatever. That's okay. It's just do the research. But if you find one that's like 60 years later, come on. So that's that. Homework number 4, I don't know when you have to submit it, so just look at your course outline. Figure it out. I can send you an email about this, or you can do the work, but just find what is the largest human gene. What is the human gene that encodes the largest protein? Now, just a little hint on this. Maybe they're not the same, okay? Splicing, all that, so think about that. What is the smallest gene that encodes a protein in base pairs and amino acids? So maybe this one is the same. Think about this. And then tell me how confident are you in your answers, okay? So this one here is just a little bonus here because I know that you'll do your research and maybe you will not be sure. So tell me how confident you are and support that by telling me what you found and what you didn't find and which one makes more sense, okay? Yes, Shauna.
How long does it have to be also in format?
Well, these will take you like 4 or 5 words. This one here, just do 100 words or something. Good question. Supported, huh?
It's not in like a question and answer form.
Yeah, you can just do the largest human gene is this. Put the reference. Smallest gene, put the reference. And then the largest protein, gene that encodes a protein, put the reference. Okay? Simple. That's it.